Hello everyone, welcome back to another video, and in this one we're going to talk about a concept from genetics which is epistasis. Now first of all, epistasis is a genic phenomenon. Now it is interaction between two different genes and is not an interaction between two different alleles. So that is why it is called a genic phenomenon. Now what epistasis means, it is masking the effect of one gene by another gene. That is, we have, let's say, two genes, and one gene is masking the effect of another gene. So this is the simple meaning of epistasis. Now epistasis can be of various different types, like dominant epistasis, recessive epistasis. So in this one, let's look at the dominant epistasis. So in case of dominant epistasis, there would be two genes, let's say A and B. These two are two different genes. So A would have the genic form of capital A, which is homozygous dominant, capital A small a, which is heterozygous, and small a. For B, it would be the same, capital B, capital B small b, small b, small b. Now, if the phenomenon is dominant epistasis, then one dominant gene would mask the effect of the other gene. Whether the next gene is dominant or recessive, that does not matter. Now, let's say if I say that A is epistatic in nature and B is hypostatic in nature. So when I say A is epistatic and the phenomenon is dominant epistasis, then whenever there is a dominant A or a capital A present in a dihybrid with capital B or small b, it does not matter, A would always mask the effect of B. So this is the meaning of dominant epistasis. Whenever there would be a dominant form of the epistatic gene, it would always mask the effect of the hypostatic gene. Now, let's say we have two parental genotypes. One is capital A, small b, and we cross it out with another genotype which is small a, capital B. Let's say these two are giving, A and B are giving two different colors. So for A, let's say it is giving purple, and since capital A is present, it would be purple. And over here, since capital A is not present and capital B is present, let's say this is giving the color red. Now if we cross it out and we get the F1 generation, that is filial 1 generation, we would get the individuals as capital A, capital B, small b. Now see, the B is the hypostatic gene, and since capital A is present, which is the epistatic gene, B won't be expressed because A is masking the effect of B, and hence all the individuals in the F1 generation would be purple in color. Now we move on and we self out the F1 generation, that is, we self cross the F1 generation. We get the F2 generation, and the F2 generation won't have that typical dihybrid cross ratio of 9:3:3:1. Instead, it would have something 12:3:1. Now, 12 would be the number of individuals, those are purple. 3 would be the number of individuals, those are red. And one individual would be there which would have neither purple nor red. Let's say it has got a color white. So this would be the case of the F2 generation ratio. Now, what can be the molecular mechanism? I mean, what can be the supposedly molecular mechanism that is working behind this dominant epistasis? So, let's say we have the gene A, which is giving an enzyme, which is enzyme A. Now, the function of enzyme A is to convert a substance, which is a white substrate. Now, enzyme A is converting the white substrate into a purple pigment. Similarly, gene B is coding for an enzyme which is enzyme B. This enzyme B is working on the white substrate and converting the white substrate into a red pigment. So that is why the gene B is giving the red pigment and gene A is giving the purple pigment. But gene A, along with giving the purple pigment, it is also doing another thing, the enzyme. It is inhibiting or suppressing the effect of enzyme B. That is why when we are having this phenotype or this genotype, then since capital A is present, capital B, although it is giving red, that is not expressed and is masked by capital A or the enzyme A that is encoded by gene A. So we can say that A suppresses, capital A suppresses B, any form of B. Now let's verify the ratio with a cross. So we said we selfed the filial 1 generation. So after selfing, we are selfing the filial 1, that is capital A, capital B, small b. We are selfing it with capital A, capital B, small b, and the gametes that we are getting. Capital A, capital B, capital A, small b, small a, capital B, small a, small b. These are the 4 gametes that we are getting. Now let's do the cross. We would have capital A, capital B, capital A, small b, capital A, small a, capital B, small a, small b. We have the same thing vertically. Capital A, capital B, capital A, small b, small a, capital B, small a, small b. Now let's do it. I'd do it and fast forward it so that we can get the results quickly. Alright, so now we have the cross, the checkerboard, and we can get the phenotypes. Now remember what I said, whenever there is a capital A, it would be purple. So let's see. This would be purple. This has a capital A, purple, 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 purple. Now this does not have a capital A. So it would take the effect of capital B and it would be red. Same case for this one. Be red. Again purple, purple. Again it does not have a capital A but a capital B. So it would be red. And since this is not having either capital A or capital B, it won't take the color of either red or purple. Instead, it would remain the white substrate as it is and would take a third color, which is white. Now, counting up the purple, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So, purple is 12. Next, coming to the red, 1, 2, 3. So, red is 3, and we have only one white. This one, 1. So we got the ratio as 12:3:1. Now, if you see that 9:3:3:1, if you add these two up, the phenotypes or the phenotypes that it was showing, if you add these two up, so this would be 12:3:1. So that is all about the dominant epistasis and how it works, what is the molecular mechanism, and what can be the change in the ratio of the whole checkerboard or the phenotypes.